You're not dead.
I already took it.
Everything is trying to kill you constantly.
Could you guys mute your mics, please? There's a couple of things I'd like to do today. The main thing is go over the worksheet that you have on how to solve Mendelian inheritance problems. So we'll do that. There were nine problems on that sheet. We'll go over as many of them as we can and make sure that everybody understands how to do them and the thought process that goes into solving these problems. So I'm going to, well, before I do that, are there any questions from anyone about anything before we start?
You already have this. Why?
Teacher, I have a question about our project that we should mention about the problem for cystic fibrosis to all organs, or just only about the pancreas or lungs?
Whatever your mutant tells you. Some mutations cause a greater effect on one organ as opposed to another. If you have several organs involved, deal with them all.
Okay. Thank you, teacher.
Okay, anything else? All right. I'm going to share my screen. Now you should be able to see the worksheet. Yes?
Yes.
Okay. So let's talk about the first problem here. In guinea pigs, black fur is dominant over white fur. How could an animal breeder test whether a black guinea pig is homozygous or heterozygous? Okay, just looking, and I want to emphasize that it's really important to read every word in this type of question very carefully, because there are key words that give you a lot of information. So just looking at that second sentence, how could an animal breeder test whether a guinea pig is homozygous or heterozygous? What does that immediately tell you about what kind of a test this is? We went over this in class. Anybody remember, if you're testing for zygosity, what's it called? Okay, well, I hope that you all went over these questions in advance so that you have some idea of how to do them, and we can just clarify any questions that you might have had. This is a test cross. The breeder is testing for zygosity. So he knows that black fur is dominant over white fur. So that means we give the colors a genetic symbol. Black is B, it's homozygous, big B, or it could be either homozygous, big B, or it could be heterozygous, big B, little b. Well, we know that the other trait is white, and it's recessive, and we know that it's homozygous, or it wouldn't be white. So we have two possibilities, and I've drawn two crude Punnett squares here, where we have the recessive little b on the left, and if the unknown is homozygous dominant, it's big B, and all of the progeny would be black. They're all heterozygotes. If, on the other hand, the unknown is heterozygous, big B, little b, then it would turn out that half of the progeny are heterozygous, and therefore they would be black, and the other half would be homozygous recessive, and they'd be white. So depending on how the test cross, what kind of progeny ratios you get, determines the genotype of the unknown parent. So is that clear to everybody, first of all? That you recognize this is a test cross, and secondly, what it tells you about the unknown parent.
Yes, it's clear.
Okay. All right, then let's go to the next one. Scott, you're very good at describing these things. Could you do number 2?
Yes, sir. Is everybody hearing me?
Yes, we hear you.
Yes.
Okay. All right. So question number 2. A short stem yellow pea pod plant is crossed with a true breeding long stem green pod pea plant. Assume that the long stems are dominant to short stems and green pods are dominant to yellow pods. They want to know 2 different things. 1, what are the genotypes and phenotypes of the F1 generation? And 2, what are the genotypes and phenotypes from an F1-F1 cross, or basically making an F2 generation? So we need to read back through this and look. This first plant, short stem, we have to figure out, is short stem dominant or recessive? And it says long stems are dominant, so therefore a short stem must be recessive. And we start to assign our variables. If you look in the blue, we have assigned capital L as a long stem and lowercase l, that's not an I, that's a lowercase l, as short stems. Then we look at the pod color. So this is going to be a dihybrid cross, because the first question was a monohybrid because we're only looking at one trait. This is a dihybrid, which means we're looking at 2 different traits at the same time, which means we could end up having to do a 16 box Punnett square, but we'll see what happens. So we're looking now at the second trait we're interested in, which is yellow, the color. And again, we read back through, it says green pods are dominant to yellow pods. So we now assign our second genotype variable for color. We use capital G for green pods, lowercase g for yellow pods. So is everybody following along as to how we've set up the variables and how we went through that process?
Yes.
Okay. So again, we read through, what do they want us to know, or what do they want us to tell them? And it says they want to know the genotypes and phenotypes of the F1 generation. What's the difference between a genotype and a phenotype?
Genetic material versus expression.
A genotype is like the actual it could be like heterozygous or homozygous, like which gametes it actually receives, but the phenotype is what it shows.
Excellent.
Like
Yes.
dominant versus recessive.
And I kind of heard something else in there just before you. It sounded about right. Genotype is concerned with the alleles and the genes, and genotypes will only consist of these variables that we're assigning. Whereas a phenotype is a descriptive word like tall, short, yellow, green, and that is a result of taking a look and seeing what traits are dominant to what kind of inheritance we've got going on, and then deciphering that to decipher the codes to give us a color or a descriptive word. So we read a little further. We know that it says we crossed a true breeding long stem green pea pod. So that's another key. True breeding. It means that this individual is homozygous, either homozygous dominant or homozygous recessive, but it always produces an identical homozygous offspring or progeny. So if this is true breeding long stem, it can only have big L as its potential alleles, or the alleles that it could put into a potential gamete. It's also green. And when they say true breeding like this, they mean for both traits. It will always breed long stem and it will always breed green. So green pea pods, if it's always green, it must be a big G, big G. So these are words to look for. If it was a true breeding short yellow, then we would know that it was homozygous recessive for both traits. It would be little g, little l, little l.
If I can add just one thing here, Scott, true breeding also means that it's an inbred. So that's another term that you have. A true breeding plant means that it has been inbred for a number of generations so that, as Scott says, it's homozygous at all loci.
Thank you, sir. Absolutely. So these are key words and key indicators that you need to be picking out of these like paragraphs they seem to give you for these types of questions. That's why it's important to know what the terms are and how they relate to the problem. So again, true breeding, it's a key indicator of a homozygous or inbred. It could be either dominant or recessive, homozygous dominant or recessive, but it's not going to be heterozygous if it is true breeding. All right, in this instance, the true breeding plant is homozygous dominant. As we said, big L, big G, big G. And the short stem yellow pod must be homozygous recessive, little l, little g, little g. It is also a true breeding plant, although it's not explicitly mentioned as such. We know that it's true breeding because we know that it must be homozygous at all loci. And in order to be short stem and yellow, it has to be little g, little l. So the problem doesn't come out and say that it's a true breeding short stem yellow, but you are supposed to be able to pick that out from looking at the genotype here. All right, so we've assigned our variables and we start working our problem. We say a long stem is dominant because that is what the problem tells us. So therefore, big L is dominant, little l is recessive. Green is dominant, big G, yellow is recessive, little g. Now we start to cross these two parents, looking at all possible gametes that can be produced. The first plant, if I come, no matter what combination I make, every answer of one of the alleles for color and one of the alleles for stem length will always come out to be big G, big L. Those are the only possible outcomes for that, for the gametes that first parent can produce. And if we look at the second parent, because it's little l, little g, the only gametes it can produce will have one little g, one little l. So this next line here says gametes, big L, big G from the first parent, little l, little g from the second parent. And those are going to be what we use in our dihybrid cross. So when we make a Punnett square of big L, big G, and little l, little g, we will find out that you will come out with 100% heterozygotes. Big L, big G. All right. That is a truism for all these Mendelian problems. If you have two homozygous parents, one homozygous dominant, one homozygous recessive, all of the offspring of that generation will be 100% heterozygotes. So those are some shortcuts you can know and use in these types of problems. You don't even have to do a Punnett square. You just know that homozygous dominant crossed with a homozygous recessive will give you 100% heterozygotes in the F1 generation. So big L, big G, little g, is that a phenotype or a genotype?
Genotype.
Genotype, yeah.
Yeah, genotype, right? You wouldn't, I mean, it sounds very awkward to say, well, this plant is big L, big G, little g. It's not descriptive. So it's a genotype. And the answer, since the question had asked us, what will be the possible genotypes? Well, we know 100% of the genotypes of the F1 offspring will be big L, big G, little g. But it also asked us about the phenotype. So now we have to refer back to our problem and decipher what the phenotype will look like. So if long stems are dominant to short stems, what will big L give me for stem length?
This one, long and green, it's a phenotype.
Yes, long and green. So the answer to the first question they asked, what are the genotypes and what are the phenotypes? We will have 100% big L, big G, little g genotypes and 100% long stem green pod plants. So now it asks us about the second, the F2, if we take an F1 and we cross it with another F1, right? Did everybody follow so far on how we answered the first portion?
Yes.
Okay, anybody that didn't? Okay, so now we move on to the second question. We want to know about this F1 cross. Well, when we cross, we're going to have a big L, big G crossed with another big L, big G, little g. And we need to figure out what gametes each of those parents can produce. I like to use something called the FOIL method. It's similar to what you learned for FOIL in math. And FOIL stands for first, outer, inner, last. And what you do is you basically say big L is one parenthesis set and big G, little g is the next parenthesis set. And you simply look at the first two things, which will be a big L, big G. Then you look at the outermost things, which will be a big L, little g. Look at the innermost things, which will be a little L, big G. And you look at the last things, which will be a little L, little g. This is a very fast way to figure out what all the possible gamete combinations will be. And as it says, use the FOIL method, gives big L, big G, big L, little g, big, or little L, big G, little L, little g. All right. We need to know all those gametes because we have to compare. In this case, we know that since we're doing two heterozygotes at both loci, each one of them can produce this particular set of potential gametes. Therefore, we have 4 options for each parent, and this does lead us to a 4x4 or 16 square Punnett square. They're no different than the small ones. It's just we have to make them a little bit bigger. Along this top axis, we've set up one parent and all the possible gametes. On the left-hand side, we've set up the other parent and all the possible gametes. Then we simply start looking at combinations. A big L with a big L gives me big L, big L. A big G with a big G gives us big G, big G. And we know that phenotype will be long-stemmed and green. We do this for each individual gamete combination. And you will come out with a table that looks something like what you see here. Now we have to go back in and figure out how many of each genotype will be produced. And you will basically just start a list. Look at the very first box and it says, okay, big L, big G, big G. Write that down and put a little tick mark next to it. Then you go to the next box, big L, big G, little g. I haven't already written that down, so I add it as a new genotype and put a tick mark. And I do that for each and every box until I come up with this following list and the numbers of each genotype that would be present. Sometimes the questions will ask you how many of each, sometimes they'll ask you the ratio. You just have to read the question. In this case, it's simply asked for the genotypes, so a listing of the genotypes would be sufficient. All right. Do we understand how this works? It's a little tedious, but do you understand how you go about figuring out all of the genotypes?
Yes.
Okay. Then the last thing we have to do is decipher the phenotypes. We've already done that in the table. So the first thing I'll do is write long and green, and I put a tick mark. And then the next one's long and green. I add a tick mark. Long and green, add another tick mark. And I keep going through, and I find here a long yellow. Well, I don't have long yellow, so I add that and put a tick mark. So every time you have something new, add it to the list. And anytime you come up with a repeat, put a tick mark in the appropriate phenotype. And what you will find is, again, this is a pretty much a rule with Mendelian dihybrids. If you have two heterozygotes, you will find that the phenotypes will always occur in a 9:3 to 3:1 ratio. So if you're working a problem with two heterozygotes in a dihybrid cross and you don't have 9:3 to 3:1, you may very well have made a mistake somewhere. But it only works for heterozygotes at both loci. All right. So the answer to this question, second part, would be that list of genotypes and that list of phenotypes. Okay.
Yeah, I have a question about these vertical and this horizontal. This is the parents' genotype.
That is correct. One parent will go horizontal,
Uh-huh.
and each box will include one of the potential gametes.
Yeah.
And the other parent will be in that vertical boxes, the column.
Mm-hmm.
And again, it will be each of their potential gametes has to be listed so that you can make an appropriate cross for it.
Yeah. Thank you.
Okay. And it doesn't matter whether you have the male or the female on the top or the bottom. You can't mix them up. Male, the male parent has to go on one of those. The female parent has to go on the other. But it doesn't matter which one you put where.
Yeah, here it's important the genotype.
Yeah.
Not the gender or sex.
Correct. We're not so worried about that. As long as one individual's gametes are listed along one axis and the other individual's gametes are listed on the other axis.
Thank you.
You're welcome. Okay, that's dihybrids.
Any other questions about this is a typical dihybrid cross. Okay, thanks, Scott. Let's go on to number 3, which.
Excuse me, teacher. I have a question. Question regarding this. It's the generation that makes. It's not that exact that every generation with this genotype, it's make this phenotype or genotype, their offspring or their children, like this. It's not the exact formula, or it's a guess for this genotype when their parents have these genotypes and they have like these genotype and phenotype offspring or children. It's the exact formula for every person, or it's a guess. It's a Mendelian.
It's absolutely not a guess. It is absolutely the only possible combinations you can get for genotypes.
Oh.
And statistically speaking, you will get the 9:3 to 3:1. Will it work out exactly that way? No, absolutely not, because there's randomness involved. But in general, you will have the largest amount of your offspring be long green. Long yellows and short greens will be roughly the same, and short yellows will be by far. If you were counting, say, 1,000 plants, you would see that the short yellows were the least number that you produced. then the long greens were the most that you produced, but it may not come out to exactly 9-3 to 3-1. This is simply using statistics to tell you what you can expect, and somewhere in that neighborhood.
Yeah.
Now for the genotypes though, that is all. That is not a guess. That is the only combinations of genotypes that can be produced. So it's the phenotypes where things may come out a little different looking.
Okay, any other questions about the dihybrid cross?
And that was a good question.
Yeah. Okay, let me go through this next one. It's interesting because it's sex-linked, X-linked. Hemophilia, known as bleeder's disease, it's recessive, and the mutation for it is on the X chromosome. So the question asks to use a Punnett square to show possible genotypes of offspring of a man who is a hemophiliac and a normal but heterozygous woman. So it says X is the normal clotting gene and X star is the gene for hemophilia. So as Scott said, we want to ask, what are the gametes here? Well, we know that the man is a hemophiliac, and we know that he is XY. And so since hemophilia is X-linked, we know that he has a mutation on his X. So the gametes that a man can produce are X star and Y. Now the female is XX, and it says that she is heterozygous. So that means that she has one normal X and one mutant X, X star. So the question asks to use a Punnett square. So you make a Punnett square, 4x4. And we have the man on the left, he's X star Y. And the woman atop, she's X, X star. So if you put them together, the fertilization possibilities, in the first box, it's X, X star, and that would be a normal female. So even though the mutation is on one of the X's, the female has another X, and this is a recessive trait, so the female will be normal, even though she's a carrier. In the next box, it's X star, it's another female, and she will be a hemophiliac because both of her X's have the mutation. Then if you go to the males, X and Y, that's a normal male. And if it's X star Y, that's a male, and it's a hemophiliac male because there's only one X in a male, and it is mutant.
So based on the question, what should your answer look like? They want you to show the possible genotypes. All you really would have to show is the X and the Y combinations. It's nice to do the phenotypes because that kind of helps, but helps you determine what's going on. But the true answer would only require you to list the genotypes.
Yeah, that's an important point. You answer the question precisely, look at what's asked, and your answer should be only addressed at that question. Okay, is everybody?
Teacher, I have a question here. There is female have hemophilia or male, because if there is female also have hemophilia, it's this is the offspring that is female and with hemophilia. If on the female partner is normal, doesn't have a mutant gene, it's possibility of all female generation have maybe hemophilic.
If the female parent,
Yeah, here is female is normal or hemophilic also. Father and mother, both parents are hemophilic, have mutant gene.
not understanding the question.
Teacher, I have a question about these parents. Male and female both have a mutant gene
Right,
and X.
yeah. Yeah,
Yeah.
the question says that the woman is heterozygous. So that means she has a normal X and a mutant X.
Okay, thank you. Yeah, just I was confused about the mother.
All right, and the question,
So, excuse me, Professor. So for the women, she has X and X. He says if it's H capital. So why we don't put H capital to the first one? Because in the question they said where X H capital contain the normal. So it will be heterozygous. One would be H small, one would be H capital for the women.
that is, it's a good question because it raises the issue of sex linkage. So when it's sex linked, since it's X and Y, you don't have large and small, large or uppercase and lowercase letters. You have only uppercase, and you indicate a mutation with some kind of a superscript. In this case, the superscript is a star. Scott, do you have a comment about that?
No, just for autosomal problems, like the first two that we worked out, you use the large and smalls. However, for sex link, we have to keep track of X and Y. And so again, as the professor said, you can use a subscript. You could say that normal is X subscript big H and X lowercase h if you wanted, but you frequently see it more conveniently written as just X, capital X, and X star. Because in these types of problems, you're typically only tracking one thing. And so just having the asterisk is easier to write and quicker.
Yeah, I'm just asking because it's mentioned in the question. They wrote H capital. It's normal.
I don't see a capital H mentioned anywhere in this question.
In the where X over H capital contains the normal Catalan genes. It's there.
Yeah,
says
we might be having an old version or something, but mine also has XH instead of X and X star.
Let me take a quick look then. Okay. So that came out of the files on Canvas or modules or
I'm not sure.
This would have been
last week.
and of course, oh, there goes Canvas. It was very slow. Mendelian genetics practice problems. Okay,
Yeah, I think I got
so
about-
yeah, I see what you're saying. So it could be used as X superscript capital H to be normal, and
Yeah.
X lowercase superscript lowercase h for hemophilia. The notation comes out exactly the same, but I see where the confusion came in. The question on the worksheet does use X superscript capital H and X superscript lowercase h.
Okay.
Okay, let's go on to the next question. It's a bit different. Scott, could you describe this?
Excuse me, Professor. There is in the previous question and the third question, the female generation or daughters are the a big risk of the hemophilia than a female generation.
You mean statistically?
The parents has mutant gene and X chromosome, and the next generation, their offspring or their children, female children is more risky than male children because they maybe have normal X with Y.
Well, you can see the answer in the Punnett square. Males and females would be equally likely to be hemophilics.
But here is the normal female. Maybe she's mutant. She's mutant. It's in genotype looks mutant gene, and maybe in the next generation of this child, maybe be dominant.
Well, yeah, the question doesn't go to the next generation. It stops with this one.
Yes, I have a question about the female children or male children. Female is in both as the risk of the mutant gene is greater than male.
No,
It's half and half.
Oh.
it will, if you combine the way the problem reads, it will always be 50-50.
Oh.
Typically though, X-linked traits affect males more than females.
Because guys only have the one X, so therefore if they have the X star in this case, there's less chance of there's no chance of a second X coming in and sort of suppressing that.
Yeah, the term they typically use is masking, but you're absolutely right. A dominant allele can mask in a recessive allele.
Yeah, thank you. Thank you, I got it.
All right, so we'll go on to number 4. Lesch-Nyhan syndrome is a metabolic defect caused by the lack of the enzyme hypoxanthine guanine phosphoribosyl transferase, which more conveniently called HGPRT. This disease is the result of an X-linked recessive mutation and results in mental retardation, self-mutilation, and early death. A normal man marries a woman who had a brother who died of LNS, Lesch-Nyhan syndrome. You are the genetic counselor who will advise them on their likelihood of having an LNS baby. So let's look at our questions. What are the possible genotypes for the woman? And then what are the possible genotypes for the man? So we have to do a little digging around. This kind of question is a puzzle question. What are the possible genotypes for the woman? Well, we know the disease is X-linked. So is this an autosomal or a sex-linked disease?
X-linked?
It's X-linked, sex-linked, right? So it's a recessive mutation. So therefore, in order to have this, a woman either needs to have 2 X mutants or a man has to have 1 X mutant. All right. So the genotype for the woman, we don't know what she is, but she had a brother who died of LNS. So
her mother, it's for sure she's carried the disease.
Her mother then did have the disease, or possibly her father, but yes.
No, father impossible, because he has only 1 X, so he will die early, so he will not get married.
Thank you. That's very important. And I was going to bring this up. You read the question, it says early death. When they say that, they're talking about somebody who's not going to survive to reproductive age. So therefore, the woman's father could not have had the disease. He would not have survived long enough to reproduce. So we know that her mother must have had the trait, right?
Yes.
So if the mother had it, did she die from it?
No, she only carried.
Right. She only carried it. So she must not be homozygous recessive. She must be a heterozygote. So we can look then at so the
we
mother
will
survived
look-
her death, so
yeah,
she was heterozygote or she was
Yep.
homozygous.
because her brother died.
hmm
That's why she carried the disease. But she might have or not have, because her mother has two eggs. So one she's carry, one she's not once. So she can get one X that it doesn't have the disease from her mother, and one X from her father. So she might be don't have any problem. But if she get the X that has a problem, and one X from her father doesn't have problems, so there is 50%. The children for girls, maybe they have the disease or they don't have it. I mean, carry or not carry, but no one of them, it will be like have two. It's only will be carry or will be doesn't have the disease.
Correct. So this woman has two possible genotypes. And we have to look at both of them. She could be X normal, or she could be X normal and X diseased, which again, we're using X star. So we have to look at both of those. Now, what is the man's genotype? So we've got that answer. The possible genotypes for the woman will be big X, or big X star. What's the man's genotype?
XY.
It must be XY, normal XY, because he's also survived to reproductive age. All right, so there's our answer for the first question. All right, we're going to say the woman, big X, or big X star. And what is the man's genotype? Must be big X, big Y. Now, the question number 2, or part B, for each of the woman's possible genotypes, what are the possible genotypes of her offspring and the implications for survival for each of them? Well, we have those listed here. We have to work out two Punnett squares. The first assumes that the woman is homozygous dominant and unaffected and not a carrier, and the man is also unaffected. So if we do our cross, we find out that we will have normal children because there's no mutant gene involved, and 50-50 for both. Half will be normal females, half will be normal males. Now we look at possibility 2, where we have to include the fact that the woman has a carrier or she's heterozygous for this condition. And if we start doing the math here, we'll see that if we look along the first, the woman is obviously on the left-hand side. So let's look at that first row. If we take her big X with the father's big X, we get a normal female. And if we take her big X with the father's Y, we get a normal male. If we look at the next row with the X star, we'll see that she will either have a normal female or we'll have a sick male. However, is that truly a normal female? Is big X star a normal female?
Carrier female.
Carrier female might even be a better answer, but she will not be affected. She will not have the disease. So that's kind of why they're saying normal. But in truth, she will be a carrier for the disease. All right. And with the male's got 1X, do we call him a carrier?
Yes.
Anyone else?
Because when you got married and the next generation, when it's female.
Well, we're not looking at we're not looking at the parent. We're looking at the offspring here. So this offspring, if it is X star, he will be sick. He will have the disease. And we know that he will die early. He will not survive to reproductive age. So the question is, do we call him a carrier? And the answer is no, we don't, because he's not a
This is
heterozygote. He's either sick or he's normal. So what the answer to this question would come out, what are the, can we scroll up just slightly? What are the possible genotypes, the implications for survival for each of them? So the possible genotypes are listed again here in the table. And we could say homozygous dominant female. If that was her, if the mother was a homozygous dominant female, no children will have LNS or be carriers. If she is a heterozygous female, no females will have LNS, but half of the females will be carriers, 1 of 2, and 1 of 2 or 50% of the sons will be afflicted with LNS. So that would be a fairly complete answer for the second part of question 4. Does everybody understand how we got there, and does it make sense?
Yes.
Okay.
Okay, Scott, let's look at the rest of the questions. Would you like to pick one that is a different basis?
Sure. Because really, up to till 9, these are pretty much things you've seen before. You just need to know what a back cross is, and for question 5, and that's in your lecture stuff. So question 6 is really just a simple monohybrid. PKU is the question 7. That is very similar to what we've just done in question 4. We're ignoring question 8 right now with the blood type. The fly is simply a dihybrid cross like number 2. So let's look at number 9. Number 9's got something a little different going on. So a farmer breeds a chicken with all white plumage. Does everybody know what plumage is?
Feather color and
Colors. There you
Yeah.
go. Excellent. So a farmer breeds a chicken with all white plumage with a chicken with all black plumage. So we have a white chicken and a black chicken being crossed. All of the F1 offspring have bluish gray plumage. So they're neither white nor are they black. When 2 of the bluish gray birds are crossed, black, white, and bluish gray offspring are produced. What is the mechanism of inheritance and the genotypes of the parents, the F1s, and the F2s? So the first part is, what is the mechanism of inheritance? That's really the first question they want answered. Well, there are 3 mechanisms of inheritance that you know. There's complete dominance, incomplete dominance, and there's codominance. So which one would this actually be? I think we have the wrong thing written down here.
Codominance?
So this should be,
yeah, it should be incomplete.
this should be incomplete dominance, because we have neither a white nor a black chicken coming out in the first group. We have a bluish gray or a mix. There's an intermediate between white and black. So think of it kind of as a gray or bluish gray. Okay. So this would be a case of incomplete dominance. If it was codominance, what would we expect to see out of the chicken colors? So if incomplete dominance means neither allele, so that's really what this means. Incomplete dominance, the dominant allele completely masks the recessive allele. In incomplete dominance, they don't, the dominant does not completely mask it. They sort of, they come to a happy agreement somewhere. But in codominance, they are both alleles are equally strong and will equally express. So what might we get for a chicken in codominance?
Like a kind of like a spotted chicken, a black and white feather.
Absolutely. So you can think of it like a Dalmatian or a zebra where it's got some black feathers, some white feathers, maybe even the feathers are, you know, black with white spots or something, but you will see both colors. So that's a great key. If you see both colors coming back in the same organism, you're probably dealing with codominance. But in this case, we don't. We see this blend. Black and white makes a grayish, so bluish gray. This is an incomplete dominance. All right. What is the mechanism of inheritance? Is answered. And the genotypes of the parents, the F1s and the F2s. All right. Well, we have to now assign our colors, our genotype colors. So we'll use big B for black, and we'll use little b for white. So a chicken with all white plumage must be, and this is the parent we're talking about, but a chicken with all white plumage must be little b, because if there was any big B in there, we would.
Excuse me.
Mhm.
I think this is number 10. We have it number 10, but I didn't use it like big B and B small because we don't have a dominant or recessive. They both have have effect. So I used only white, and then the other one I used white and like marker on it for black, because we will have gray. So gray, it's not white or black. It's like mixed between them. There's no dominant or recessive of one of the trait.
That's incorrect. There is a dominant and there is a recessive. It's just that in incomplete dominance, instead of the dominant allele completely masking the recessive allele, they come to a sort of a midpoint. They come to a kind of an agreement in the middle, which is why it comes out gray. Because both black and white are there, neither one is actually masking the other. They are both being expressed at the same time as an intermediate shade in this case. So you would still be looking at just a color using either big B or little b, or if you wanted to use white, you could use big W, little w, whatever makes sense to you. The letter doesn't matter, but there is a dominant and a recessive coloration here.
So you are saying the chicken, the feather, they will have black or white, but because they are mixed, they look like gray, right?
Well, the parents, we only have one that is all white and we only have one that is all black. So we know that those traits are there somehow.
So they are there. So there's no dominant or recessive. They are there. The black give you the feathers, they are black, and the white, they give you the white feathers. So because they are mixed, because when you put them together, it's look like gray. So it's like mix the feather that's make that color, but they have white and black. So I don't think there is dominant and recessive.
I'm going to disagree with you, and I'm going to go back to saying there is definitely a dominant and there's definitely a recessive. So let's take a look though at our genotypes. It doesn't tell us whether white or black is the dominant or the recessive. So we don't know that, and the question doesn't really ask us to identify which one, but we have to account for there is a dominant and there is a recessive. But so I just picked here that
so just let me say something. If you're correct, so B capital, B small, it has to be black, don't has to be gray, because the B, if it's dominant, so we will have B capital, B small, it has to be black. We don't have to
But
say
that's
it's
the
gray.
point of the question. It's not dominant and it's not recessive in the F1. It's incomplete dominance.
Yeah, that's what I'm saying. It's incomplete. That's why we are so B, it's not dominant on B small.
Yeah, that's what this question is trying to bring out, is that their allele or genes can have dominance, or they can have incomplete dominance. And this
Yeah.
one is trying to illustrate incomplete.
Yeah, for if we said that, so B capital, B small we have to say this is the phenotype here. We have to say it's black. We don't have to say it's gray.
But that's
So
the point.
you're
that-
jumping ahead to the F1 generation, I think. We're not at that point yet. We're still trying to figure out what the parents are. Okay, are you looking at the F1s?
I'm just looking to the trait. There is no black and white. There is no one dominant and one recessive. They both have the same trunk.
If a chicken is all white, there must not be a small, or there must not be a large B in there anywhere. So a chicken that is white, based on the lettering we've chosen for big B being dominant, little b being recessive. And in this case, we're assuming white is recessive. So in order to have white, I have to have two small b's. And in order for that parent chicken to be all black, they must have two capital B's. They cannot be heterozygotes, and you're correct about that. They either are homozygous dominant or homozygous recessive. In this case, we just chose recessive as white because the question isn't explicit about which way it goes. However, that doesn't matter. Whether we chose white as dominant or black as dominant doesn't affect the answer that we come out with. So we know that the white chicken is, in this case, little b, and the black chicken must be big B, big B. And we also know that if a homozygous dominant is crossed with a homozygous recessive, 100% of our F1 generation will be heterozygotes, in which we see this, all of the F1 are blue-gray, which must mean that a big B, little b crossed, and we produced 100% big B, little b's. And because this is incomplete dominance, they all show up as gray.
Yeah, because it's incomplete.
Right. Absolutely. And so then it wants to know about F2s. So then we say, okay, F2s, well, that's a cross between F1 and an F1. So that's where this little Punnett square is, where we have a heterozygote crossed with a heterozygote. And we see that we will get one black, big B, two gray, big B, little b heterozygotes, and we will get one white, little b, homozygous recessive. And again, you see this 1 to 2 to 1 ratio when you're crossing heterozygotes. So again, we say mechanism of inheritance, it should actually be incomplete dominance. And so this is our answer. Incomplete dominance is the mechanism of inheritance, and the genotypes of the parents must have been big B, little b. The genotypes of the F1s are 100% big B, little b. And in the F2s, the genotypes produced are big B, little b, and little b. And you're right, this is question 10 in yours. Question 9 here, because this one doesn't have the blood typing. So that's why it's off by 1. Did this make sense to people? The discussion kind of got a little involved, but the basics of it are not really anything different than what you saw in, say, question number 1. It's just a simple monohybrid. The difference was the mechanism of inheritance.
Seemed to make sense.
Okay, so it's important to be able to recognize what the question may be telling you about mechanism of inheritance. And in general, if you don't see anything that indicates a mechanism of inheritance, that you will assume complete dominance unless you see something that indicates a different type of inheritance is at work.
Okay, we're running short on time. So Scott, is there another question here that hasn't been dealt with that makes another point?
Well, question 5, we just get a back cross. And in a back cross, you just have to pay attention. You're going to work it out. This is a monohybrid cross. We're looking at colors only, red to yellow. But we have to pay attention to the directions it's giving us. For instance, 5B. We're taking the offspring of a cross of the F1s back to the red parent. So we actually have to figure out what the cross of an F1 would be.
Now, the distinction here is that instead of going to an F2 where you cross F1 by F1, the F1s are being back crossed back to a parent rather than to a sibling.
So in a way, you could think of it as an F1.5.
I guess you could say that.
You would never see anybody say that, but yes, they would be asking about a back cross, and they would have to specify which parent you were back crossing to. Because if they didn't, you'd have to assume, you'd have to work out 2 possibilities, one for each parent. But so this is really, I think, the only one that's a little different.
Question 8 is different. Just I want to confirm if my answer is correct or not, because I did it's for blood.
Yeah, the blood question was
Yes.
removed from this set.
Oh, so they are they correct? Oh, you don't have it?
No, it's not on here because it was removed from this particular question
Yeah,
set.
I'm sorry, I sent the wrong problem set to you.
Oh,
This is the updated one.
If you would like to know about that one, absolutely, I can work it out with you. Come to workshop and we will work it out.
yeah, I just want to know if my answer is correct. So if they say the parents are they correct?
So hang on a
They are
second.
not.
What did I just say? We don't have it here. It will confuse
okay.
a lot of other people because they can't see it because it's not here laid out. So I don't have the answer right here. I would have to work it out real quick. I can't give it to you. So I can't tell you whether you're correct or not right now. Give me a few minutes and I'll be able to, but I can't at this moment.
Okay.
So let's let the professor go ahead and wrap things up while I'm working on this. If I get you an answer before the end of class, great. If not, please stop into workshop and I'll give you the answer for that, or I can put it in the discussions.
Okay.
Thank you.
All right. I want to remind you that on Wednesday, there is a quiz. That quiz will cover cell division and Mendelian inheritance. In other words, the kind of material we've just been going over today will be about half of the quiz on Wednesday. The other half will be on meiosis and mitosis.
Teacher, this quiz not include meiosis and mitosis?
The quiz will include meiosis and mitosis.
Okay.
It will be like all other quizzes, 15 minutes. And I think there are 6 questions on it. So it's fairly short.
Professor,
Yes?
this is Mary Kip. How do I get to the students who are in my group?
Have you sent them emails?
Yes, I think I have.
I'm having a hard time hearing you. Are you speaking into your microphone?
Yes.
Okay, speak loudly.
Okay. I have 2 students with me.
Okay, are you finding that the other members of your group are unresponsive?
I have the name, but I don't know how can I get it.
You should be able to get their email addresses from WebAdvisor.
You can also just directly message them through Canvas.
Just by putting their names in.
Correct.
Thank you.
Yeah, you raise a good point, though. If you find in your group that you have a member that doesn't respond to your inquiries, let me know, and I'll try to find out why you're not getting a response. And if there is someone who just does not want to participate, they don't have to. And if there are a significant number of these, we can make rearrangements among the groups. But first, make every effort to contact the other members of your group and make a plan for how you're going to approach this project.
So the answer to Agared's question is the parents are not correct.
Oh, yes, that time I did. So because they are heterozygous, right?
Well,
Because they are
they-
A and B.
they
So-
must be heterozygotes, both
Yeah.
of them. Yes.
Yeah, okay. Thank you.
You're welcome.
Professor, just
Yeah.
I don't know if this is a good time, but it was just about the groups. I'm not, I don't think they're here again, but I noticed that like both my group members haven't been in class they weren't there last time either. I haven't reached out to them yet. Will that affect the project if they're not, like, are they gonna be missing stuff in class that we need for the project?
Yeah, when I made these groups, it was based on the roster as of that day, the day that I made the groups. And so I did not do it, or did not take into consideration how active an individual might have been in the class. So if you, I would say, first of all, reach out to the other group members, and if they want to be involved, fine,
Mhm.
go ahead with them. But if they don't, let me know, and I
Okay.
will investigate further, and we may have to do an adjustment of the groups.
Okay, no worries. Yeah, I'll reach out to them and see what.
And as I say, if you don't get a response,
Mhm.
get a negative response, let me know.
Okay. All right. Thank you, Professor.
Professor, I have a quick question. For Wednesday, we'll be starting natural selection and evolution.
Yeah,
Alrighty, thank you.
yeah, that will be the topic. We'll switch. That sort of follows on Mendelian inheritance. Yeah, so we'll talk about natural selection and evolution on Wednesday. That will be a short lecture period because I'll have a quiz. Okay, anything else? If not, that's it for today. And I will see you on Wednesday.
Have a good one.
Okay,
Thank you, teacher. Have a-
end the session.
Thank you, Professor. Have a good day.
Okay, you too. Bye.
Thank you. Bye.